Bismillahirrahmanirrahim. Assalamu alaikum dear students. Welcome to your channel Anatomy Canvas. Today's class of histology we are going to discuss with the histology of the nervous system. Learning objective for the today's class are to describe the histological features of the cerebellar cortex, to describe all the cell types present in cerebellar cortex in detail, and to describe the histological features of the spinal cord. So cerebellum, the little bit about its gross anatomy. It is the largest part of the hindbrain. As you can see, this is the largest part of the hindbrain. It weighs about 150 grams. It composed of the many folds called the gyri or the folia, and these folds have outer cortical gray matter with inner medullary white matter. So histologically, the cortical gray matter is divided into three main regions. Most external is the molecular layer. So this outer is the molecular layer, and it is mainly a synaptic layer with a few pericardia and many unmyelinated nerve fibers. The types of the cells which are present in this outer layer are the stellate cells and the basket cells. So basically it is a synaptic layer. It contains very few pericardia. Most of the time, most of this layer is formed by the synapses of the dendrites and the axons. The next, the stellate cells which are present in the molecular layer, these are the star-shaped cells. They have many short dendrites and fine unmyelinated axon, and their axon have their axon synapses with the dendrite of the Purkinje fiber. So these are the dendrites of the Purkinje fiber, and here is the synapses of the axon of the stellate cell with that of the dendrites of the Purkinje fiber. Next cells are the basket cells. Here you can see the basket cells which are present. They have many dendrites and an unmyelinated axon, and these unmyelinated axon run horizontally and give terminal arborization around the either body of the Purkinje cells or the dendrites of the Purkinje cells. So these two types of the cells, the stellate and the basket cells which are present in molecular layer, their axons have arborization, and their axons have synapses with the dendrites of the Purkinje cells. Next is the middle cell layer, and this middle cell layer is formed by the Purkinje cells. It is the neuronal cell layer. So single layer of the neurons with pericardia lying between the molecular layer and the granular layer. So these are the single row of the Purkinje cells which are present over here. These Purkinje cells are flask-shaped cells. They have dendrites which run into molecular layer where they synapses with the axons of the stellate cells and the basket cells, and they have a long axon which is usually myelinated, and it crosses the granular layer and enters into the white matter. It is the main outflow fiber of the cerebellar cortex. Next is the inner granular layer, and this inner granular layer have two types of the cells. Most abundant of these are the granule cells. These are smallest neurons in the human body, having a size of about 5 micron meter diameter. These neurons are multipolar with 3 to 6 dendrites and the one axon. So here you can see many dendrites, 3 to 6 dendrites of these granule cells, and one axon which run into the molecular layer, divide and into a T-shaped manner, and it then synapses with the dendrites of the Purkinje cells. Next type of the cell which is present in the granular layer is the Golgi type 2 cell, which is also an inhibitory cell. It has many dendrites and axons which enters into the molecular layer where they also synapses with the different cells. So two types of the cells are present in granular layer. These are the granule cells and the Golgi cells. One types of the cells present in the middle layer, that is Purkinje cell layer, and these cells are Purkinje cells. And two types of the cells which are present in the granular layer and the molecular layer. These are the stellate cells and the basket cells. Now have a look at this diagram. This is the basically inferior olivary nucleus having its output into the and having synapses with the Purkinje cells. And these are the stellate cells and these are the basket cells, and axons of both of these cells having synapses either with the dendrite of the Purkinje cells or with the cell body of the Purkinje cell. Here you can see the Golgi type 2 cell which is present in addition to the granular cell, and also you can see these are the mossy fiber basically coming from the spinothalamic or the other regions of the cerebellar cortex, and these mossy fibers synapses with the cell body of the granular cells, and here they form the glomeruli. So different the cerebellar cortex and types of the cells which are present. The molecular cell having a molecular layer having dendrites of the Purkinje cells, the axons of the granule cells, the climbing fibers, the stellate cells, and the basket cells. Next is the Purkinje cell layer, which is having Purkinje cell, the biggest cells in the central nervous system. That is also a main identification point in histological slides for the cerebellum. They have a conspicuous cell body and their dendrites are highly developed, assuming the aspect of the fan, and axon project to the cerebellar nuclei. Third, granular cell layer contain the cell bodies of the granule cells. They receive excitatory input from the mossy fibers. Each axon send an each cell send an axon to the molecular layer where it bifurcates to form the parallel fibers, and these fibers form the excitatory synapses on dendrites of the Purkinje cells. Other types of cells which are present in granular cells are the Golgi type 2 cells. So this is the H and E stain section of the cerebellum, in which you can see from in this slide from this area to this area, this is the cerebellar cortex, and out of this cerebellar cortex you can see this outer area is the molecular layer, then this is the Purkinje cell layer, and then this is the granular cell layer, and then we will label it with another color. So this is the white matter which is present in the center of the folium, and this whole is the folium. So you can see in this slide, this is the outer cortical area which is formed by the outer outer is the molecular layer, then is the Purkinje cell layer, and then is the granular layer, and inner is the white matter which is mainly containing the axons, either input or output of the cerebellar cortex. So this is the slide of the cerebellum at higher magnification, in which you can see this outer area is the molecular layer, then and this area is the Purkinje cell layer, and this is this all of it is the granular cell layer. And in this molecular cell layer, two types of the cells are present. These are the stellate cells and the basket cell. So you can differentiate between the two types of the cells, that the basket cells having a large amount of the cytoplasm. That's why around the nucleus you can see a clear area to differentiate this basket cell from the from the stellate cell. So two types of cells are present. Mainly this is a synaptic layer. Then look at this area. In this you can see the only type of the cells which are present in the Purkinje cell layer are the Purkinje cells, and you can see this is a flask shaped cell in which prominent nucleus and nucleolus is present. So this is the Purkinje cell in which you can see prominent nucleus and nucleolus is present, and it is a flask shaped cell. Then this whole is the granular cell layer, and in this granular cell layer again two types of the cells are present. The main population of the cell are the granule cell, and in addition to granule cell you can differentiate. You can see these cells which are present in addition to this. I first highlight it. This type of the cell which is present in addition to these granule cells having larger amount of cytoplasm. So these cells are the Golgi type 2 cells which are present in granular layer in addition to the granular layer. So these are the different types of the cells which are present in the cerebellar cortex. Now, the Purkinje cells are having excitatory input, the mossy fibers to granule cells to the parallel fiber to synapse on the Purkinje cells. Vast combinations provide sensory context. Interneurons for the Golgi cells are interneuron for the granule cells, and basket and stellate cells are interneurons for the Purkinje cells. These are again another H&E stain section of the cerebellar cortex. This is one is the lower magnification and this is the higher magnification. In this magnification you can see this outer is the cortex, that is the gray matter, and this inner is the white matter. So in this outer cortex you can see this outer zone is molecular layer, then the Purkinje cell layer, and then the granular layer. This is same slide at higher magnification, in which you can see this is the molecular layer, then the Purkinje cell layer, and then the granular cell layer. So now we move on to the spinal cord. The spinal cord, the neurons are larger and multipolar, and especially in anterior horn where large motor neurons are found. This is the section of the spinal cord. The arrangement of the gray matter and white matter in spinal cord is opposite to that of the cerebral cortex and cerebellar cortex. So in case of the spinal cord, the gray matter is present in the center in the shape of H or a butterfly shaped area, forming the anterior horn, the posterior horn, and the central area which is surrounding the central canal, forming the commissures, and outer to this all is the white matter. So from externally you can see this is the posterior median sulcus and this one is anterior median fissure. Then this is the anterior gray matter and this is the anterior white matter. This is the lateral white column and this is the posterior white column. Similarly, this is the posterior or the dorsal horn of the spinal cord and anterior or ventral horn of the spinal cord, and this smaller is the central canal, and the area of the cortex, the gray matter which is surrounding the cortex, is called anterior and posterior gray commissures. So in this slide also you can see the neuronal cell bodies which are present. Smaller neurons, smaller nuclei are there, those of the glial cell nucleus. In addition, you can also see the nerve cell fibers which are present as well. So now again have a look at this diagram. This is the anterior view and this is the posterior view. So this spinal cord is covered by the three meninges which are from outer to inner side are the dura mater, the arachnoid matter, and the pia mater, and this pia mater is closely adherent and attached to the spinal cord and it enters into the sulcus, that is anterior median sulcus. Posteriorly you can see the posterior median fissure over here. In the center you can see the central canal. This butterfly shaped area is the gray matter which has having the dorsal and the ventral horns, and this is the white matter which is anterior white matter, the lateral white matter, and the posterior white matter. Here you can see the dorsal roots which are coming from the connecting the dorsal horn of the spinal cord, and these are the ventral root. Here you can see the dorsal root ganglia is present, in which the neuronal cell bodies of the sensory neurons are present, and their central process enter through the dorsal root into the dorsal horn of the spinal cord. The peripheral process of these cells of dorsal root ganglia then combine with the ventral branch to form the spinal nerve. So this is another section in which you can clearly see the different parts, that is the anterior median sulcus, the posterior median fissure, the butterfly shaped gray matter forming the anterior horn, posterior horn, and in areas where the thoracic region, in the thoracic region where sympathetic outflow is there, you will also see the lateral horn as well. So these are the dorsal nerve roots and these are the ventral nerve roots. This is the dorsal root ganglion which is present. Again, spinal cord detail histology, the cross section of the spinal cord in the transition between gray matter below and white matter. In this you can see this is the gray matter and this area is the white matter. The gray matter contains the neuronal cell bodies and abundant cell processes, and whereas white matter contains mainly the neurons of the glial cells. And the nerve fibers where myelin sheath is dissolved in histological process. In this slide you can also see this is the main anterior motor neuron, in which you can see very clearly the nucleus with the prominent nucleolus giving an owl's eye appearance. Also the nasal granules are present, which are very prominent in this. This is another section of the neuron which is present in the gray matter. So a section of the spinal cord showing the meshwork of the cell neurons. These are the neurons and these are the glial cells, and in addition you can see the myelinated and unmyelinated axons over here. So in this neuron you can see a very clear neuron nucleus of the neuron with the clear nucleolus giving an owl's eye appearance, and nasal granules are also present and some dendrites which are coming out of the body. Spinal cord, again you can see the gray matter, it is a shape of an H or a butterfly shape. Central canal lined by the ependymal cell. These are simple cuboidal cell. These are the anterior horn. These are motor horn containing larger cells. These are called anterior motor neuron. This one is the posterior horn, which contain the which is sensory in nature mainly, and the neuronal cell bodies are located in the dorsal root ganglia, which enter through dorsal nerve roots into the dorsal horn. And this outer is the white matter. This white matter is called posterior white matter, also called the posterior funiculus. This is the lateral white matter, also called lateral funiculus, and this is the anterior white matter called the anterior funiculus. So dear students, here I complete my today's class. The references for today's class are the Basic Histology for Laikus and Siddiq, Histology Text and Atlas by Jan Quara, and Histology Text and Atlas by Michael H. Ross. This was also the last class of the histology of the central nervous system, and thank you very much for your attention for today's class. Allah Hafiz.